Assalamu alaikum everyone. Welcome to a new session in online physiology, and today we are going to discuss what we call leukemias. Okay? To begin with, by definition, leukemia is an excessive and abnormal increase in the number of circulating white blood cells, that is due to abnormal proliferation of their precursors in the bone marrow. Okay? So that is something truly life-threatening. So to be able to understand the concept of leukemias, we need to understand the stem cells from where they originate. Okay, we have stem cells called pluripotential hematopoietic stem cells in the bone marrow. Okay, all the blood cells in the circulation is derived from those pluripotential hematopoietic stem cells. They are not only fertile, but they are versatile as well in that aspect that they can give rise to essentially any blood cell type. Okay, those pluripotential hematopoietic stem cells continuously divide and proliferate to not only to renew their own population for later use, but they give rise to stem cells of other types. And there are two major types of such stem cells. We have the lymphoid stem cells and we have the myeloid stem cells, labeled here as colony forming unit spleen. Okay, the lymphoid stem cells gave rise to all the B and T lymphocytes in circulation, while the myeloid stem cells are a bit more versatile as compared to the lymphoid stem cells. They give rise to specific colonies: colony forming unit erythrocyte, colony forming unit granulocyte and monocyte, and colony forming unit megakaryocyte. Okay, so while at this stage, when we call them pluripotential hematopoietic stem cells, they can give rise to any cell type. Okay, but once they form colonies, they become lineage specific. Okay, we also call them committed stem cells. Once at this level, like this precursor or progenitor can only produce red blood cells. I'm talking about the colony forming unit erythrocyte. Similarly, the colony forming unit granulocyte and monocyte will only give rise to the monocytes in circulation and granulocytes. Okay, here I want to add, granulocyte is a fancy word for all those circulating neutrophils, basophils and eosinophils. Okay, plus basophils. And this is because all of them have one thing in common. When we look at them under microscope, they have a granular cytoplasm, and this is why we call them granulocytes. Similarly, the colony forming unit megakaryocyte gives rise to all the platelets in circulation. Okay? One thing to remember, once we have colony forming units, at this stage they become lineage specific. They become committed. This precursor cannot give rise to RBCs or any other cell type, though they are now committed. Okay, well, these cells, the colony forming units, the cell precursors, divide continuously to provide a continuous supply of all these cells in the circulation, and that varies from case to case. For example, the red blood cells on average live for 120 days, while the platelets have a much shorter lifespan, that is just 5 to 10 days on average. Okay, so depending upon their rate of destruction in peripheral circulation, be it physiological, be it pathological, the bone marrow has to offer a rate of production that equals their rate of destruction. Okay, and this whole process is influenced by what we call growth inducers and differentiation inducers. The names are self-explanatory. For example, the growth inducers predominantly determine the rate of production. For example, in the case of red blood cells, you might have heard of erythropoietin. We have so many others as well, but erythropoietin is a growth inducer for red blood cells. Okay. Similarly, there are differentiation inducers. Now here I need to explain what is differentiation. Okay. While the stem cells form colony forming units, Each and every cell that is finally produced passes through a certain process. It evolves from immature form to a more mature and functionally stable cell. Okay, so that finally each and every cell resembles exactly its parent. This is what how we call like father, like son. Okay, but these are and I need to mention the growth inducers and differentiation inducers are finally regulated at the level of genes. This whole process of growth and differentiation is strictly regulated at the level of genes. Okay, but we have a problem with all the rapidly dividing cells. If they divide rapidly, they are prone to make mistakes. The process may go erratic. This is like during photocopying, the photostat machine may produce some copies which are not acceptable. They have those gray shady lines and they are not acceptable. So similarly, during this rapid growth and cell division, we get some copies which are not acceptable. They are abnormal, they are monstrous, and you may call them cancerous. Okay, so while growth inducers and differentiation inducers are regulated at the level of genes, we have one more level of protection. We have certain genes which will get rid of those abnormal clones of cells soon after they are formed. They will be suppressed, and these genes are what we call tumor suppressor genes. They will detect and suppress every abnormal single clone of cell early in its development, so that the body has protection against cancers. Okay, I have mentioned few for reference here. We have the TP53, the RB genes, the ENC4 ARF family, and the PML genes, protecting against cancerous production of abnormal clones of cells. Okay, because once we have an abnormal clone of cell, it will produce abnormal cells like itself. 8 times those abnormal cells somehow escape suppression by the genes. We get abnormal clones of cells that divide continuously, rapidly, and in uncontrolled fashion, and they escape that suppression by the tumor suppressor genes. So as a result, we get cells that divide and redivide in an uncontrolled fashion. So here I want to mention, growth is a normal phenomena. So far it is regulated, so far it is under control. The moment growth becomes uncontrolled, we call it cancer. So during this process, there is a possibility for these cells to give abnormal clones of cells, to give rise to abnormal clone of cells, which lead to leukemias. Okay. Well, to begin with the types, as we mentioned earlier, all those cells arise either from lymphoid stem cells or the myeloid stem cells. So based on their site of origin, we either have myeloid leukemias or lymphoid leukemias. They arise here in the myeloid stem cells, we call them myeloid leukemias. If they do arise in the lymphogenous stem cells, we call them lymphogenous leukemias. Okay. Secondly, based on their mode of onset, or rather rapidity of onset, how quickly they manifest, they are either acute or chronic. Now, in the case of acute leukemia, it has a stormy onset. Say, for example, the patient is unwell for a couple of days or even weeks, and they go to hospital. After investigation, they come to know they have an underlying leukemia. Okay, so the onset here is going to be very stormy, so spanning over days to weeks at most. Okay, the chronic leukemias are more insidious in onset. Okay, so they develop very slowly, and it usually takes years for them to be noticed. Like in case of B cell leukemias, usually it takes more than 15 to 20 years for them to be noticed. Okay, they don't cause considerable problems at all. Okay, secondly, what we are concerned about what they produce. In acute leukemias, we get cell types which are not mature. Okay, so they are neither monocytes, they are nor granulocytes. Rather, they are of some, most of the times, they are of some intermediate type. They are poorly differentiated. Like we said earlier, like father, like son. Here in this case, they don't resemble the parent cells. They are a bit different and they are immature. Okay, so the second feature with acute leukemias usually, generally, but not always, is poor differentiation. Okay, we get poorly differentiated cells. Okay, while the chronic leukemias in general, they tend to produce more and more mature cells. We can exactly pinpoint whether the abnormal cell in circulation is a granulocyte, a T lymphocyte, or some other type. Okay, we can exactly pinpoint the type. So they are more differentiated in general. So these are the basic types.